Have you ever heard the term that this baby is not having both testes in his scrotum? After birth, this baby is not having both testes in his scrotum. Have you listened to this phenomenon? After the baby is delivered, baby is delivered, he is given a swab. And thereafter, his body from head to foot is observed. All right. Whether there is any congenital anomaly present or not, of which the inguinal region with absence of testes either in one scrotum or both, it is an important anomaly. As well as the baby is observed for cleft lip and cleft palate. Baby is observed for cleft lip and cleft palate because the baby will be given feeding, and if there is cleft palate, the feeding will come out from the nose, and sometimes baby can aspirate. So these are the common anomalies that are searched for after the birth of the baby. Now, our genital system is developed for the maintenance and propagation of our sex cells. There are two sources of sex cells in human being. That is there is the male sex cells and the female sex cells. Now the gonads at first remains undifferentiated. That is, you cannot tell it that it is a male gonad or a female gonad until and unless there is histological differentiation within the gonadal tissue and formation of ability to form the seminiferous tubules as well as the sex cells, which occurs by 7th week. And the primitive sex cells that comes from the dorsal wall of the gut at the mesentery, which migrates to the genital ridge by 5th week. So from 5th week, the dorsal wall of the gut is giving rise to the primitive sex cells that is passing through the dorsal wall of the mesentery and is entering into the primitive genital region. If it enters into the cortex, then the baby will have cure. If these sex cells, primordial sex cells, enters into the cortex of the baby, cortex of the genital region, then it will form a ovary and the baby will be a female one. And if it enters into the medulla, then the genital region will be directed to turn into testes. Alright, this differentiation by testis, ovary becomes evident by 8th to 9th week, and by 7th week, testis, histological differentiation of testis is complete. Right? We observe gonadal sex, then the gonadal tissue that gives rise to the testis or the ovary. Then there is ductular system of the testis, that is from the remnants of the mesonephric tubules and mesonephric duct. From the remnants of the mesonephric tubules and mesonephric duct, the ductular system of the testis develops. Following that, there are some accessory sex glands, that is the prostate and the Bartholin glands. Alright, this duct also comes from the mesonephric duct. This also comes from the mesonephric duct. And following the development of the ductular system, there is efferent ductules of testis. Thereafter comes the retitestis, efferent ductules of testis. Then comes the canal of epidermis, vas deferens, then the seminal vesicle, common ejaculatory duct, and up to the prostate. And thereafter we see the canalization of the seminiferous tubule. From where all the male germ cells comes out. Right? Now, we have to look for also the hormonal support in such development. From the interstitial cells of Leydig. Actually, we see three types of cells within the developing testis. Number one, the germ cells. Number two, the supporting cells of Sertoli, and the interstitial cells of Leydig. These three histologically differentiated cells can be seen within the developing testis. Another thing is, how can you differentiate the germ cells within the developing testis? The things are, the germ cells are larger in size with much more alkaline phosphatate and glycogen within these cells. So you will be able to identify histologically that these cells are germ cells because of excess alkaline phosphatase content and the glycogen content within these germ cells, a large size. This gives rise to the identification of the germ cells. Now, at first, the germ cells, when the germ cells enters, there is formation of reticords, uncanalized reticords. And when the epithelial cells, there is a mesothelial cell from the genital mesonephric ridge enters into this cord, canalized cord are separated from the surface. At first there was uncanalized reticord, and after that there is canalized reticord. After this canalization of this reticular tissue, that is the plexus of clusters of cells, there will be successive canalization, that is canalization of the reticord, the efferent ductules, then the canal of epididymis, then the vas deferens, seminal vesi and seminal vesicle. All these will become canalized and help in the spermatogenesis. Now the second red picture is the separation of the reticord, that is canalized reticord, and the first one is the uncanalized reticord. Alright, can you label this diagram, understand the labeling of this diagram? So the primitive sex cells coming from the dorsal mesentery entering into the medulla to form the testes, and if these primitive sex cells enter into the cortex, then it will be an ovary. By 5th week, these cells appear from the dorsal mesentery, and by 7th week, there is histological differentiation in case of testes, and by 8th to 9th week, there is histological differentiation of the ovary. We can see it, all right. And then phenotypic sex, the question of phenotypic sex comes. The phenotypic sex means the male characteristics that needs to be developed. Male characteristics that needs to be developed. Starting from the male type of urethra, the penile part, the scrotum, these all are, and of course the musculature, male type of musculature, all these are phenotypic sex. So we look for, what we look for? Number one is the chromosomal sex, next the gonadal sex, next the phenotypic sex, right? In describing the development, what will you say? That there is, at first we look for chromosomal sex, alright. If Y-bearing sperm, if any Y-bearing sperm, fertilization, egg-bearing sperm, so the combination will be XY. Now this active part of this combination is the Y chromosome. This Y chromosome, the HY locus, you know the meaning of locus in genetics, in relation to genetics? What is locus in genetics? HY locus, you should read it. HY locus, there is the histocompatibility antigen for Y chromosome that makes the surface protein of the male tissue. This HY locus positive, that is Y chromosome positive, and the pericentric region of Y chromosome gives rise to the formation of testes. Pericentric region of the Y chromosome gives rise to the seminiferous tube. The long arm of Y chromosome gives rise to the development of testes. Long arm of Y chromosome gives rise to development of the testes, right? And in case of female, absence of active Y chromosome as well as the presence of X chromosome, 2 X chromosome, that gives rise to the development of the ovary, and there is regression of the medulla. In case of male, there is regression of the cortex, and in case of female, there is regression of the medulla, right? Now, this phenotypic changeover in case of male occurs through some hormonal factor, like from the Sertoli cells comes the Mullerian regression factor. Mullerian regression factor from Sertoli cells, and steroid hormones comes from the interstitial cells of Leydig. Mullerian degeneration, Mullerian regression means the mesonephric duct that was present within the ductular system of the testis is now getting regressed, and the steroid hormone that comes from the interstitial cells of Leydig, that is the testosterone and the dihydrotestosterone. Testosterone will definitely develop the male type, give rise to the male type of development, that is the phenotypic sex will become male in nature, and the dihydrotestosterone can be converted. Dihydrotestosterone gives the continuation of the spermatogenesis. Dihydrotestosterone gives the continuation of the spermatogenesis. And from dihydrotestosterone to testosterone, it can be converted by an enzyme, that is the 5-alpha reductase. All right. In testicular feminization syndrome, there is testis. There is male pattern of body development, but there is the person is sterile due to the absence of the protein receptor that is needed for the testosterone and dihydrotestosterone that is absent in the cell. So in spite of having testosterone, dihydrotestosterone, Mullerian regression factor, due to the absence of this protein receptor, these cells cannot act as a male spermatogenic cells, and the person becomes sterile. This is called testicular feminization syndrome. Means the baby is, or the child is having all the factors for developing into testis, but the protein receptors that will be worked through the testosterone or the dihydrotestosterone is absent. This makes the baby sterile. All right. Now, after the formation of this canalization of the reticord, this is detached from the surface epithelium. This surface epithelium comes from the mesonephric ridge, and insinuates in between the canalized reticord. They are made separate, and this cells remains in the medulla of the testis. Now you can see in that picture that the mesonephric ridges on either side, they are the genital ridge in relation to its middle two fold, and in the upper part there is the suspensory ligament, and from the lower part, that is the gubernaculum. From the upper part there is the suspensory ligament, and from the lower part there is the gubernaculum. This gubernaculum goes to be attached to the genital swelling, and the middle diagram is the urogenital sinus with the 4 tubes. Urogenital sinus with the 4 tubes. 2 mesonephric tubes at the center, and 2 paramesonephric center tubes on the outside. When the gubernaculum is developing and passing through the anterior abdominal wall, there is an inguinal fold. The gubernaculum passes through this inguinal fold. That is, the inguinal canal is passed through, and this gubernaculum is attached to the genital fold that gives rise to this attaches to the genital swelling that gives rise to the scrotum, and the part of gubernaculum is attached to the base of the scrotum. These proximally, the gubernaculum has three processes. One that attaches with the processa succus vaginalis. The upper one, the peritoneal pouch, behind which the genital, the developing testis remains, is the succus vaginalis. And the upper part of the gubernaculum is attached, number one, to the peritoneum of succus vaginalis. Number two is to the mesonephric duct. And number three, to the lower end of the developing testis. And the distal part gives rise to the few extensions. Number one, that goes to the saphenous opening, one that goes to the base of the scrotum, one part that goes to the perineum, another part that goes to the anterior superior iliac spine, in front of the anterior superior iliac spine. Another one that comes before the symphysis pubis. Alright, all these are called the tail of, these are called tail of locum. These are called tail of locum. So, this garter-like extension of the gubernaculum and later the contraction that makes the gubernaculum the hemspan or the guide for descent of testis. So there is the factors like the uncurling of the fetal curvature. Number two is the contraction of this gubernaculum tissue, increased intra-abdominal pressure, lowering of the temperature. All these factors causes the descent of the testis into the scrotum. So after birth of the baby, we check the both sides of the scrotum whether testis is there or not. If it is found within the inguinal ligament, that is, testis comes in the tapering by 4th month and crosses the inguinal canal by 7th to 8th month and passes the superficial inguinal ring and comes to the scrotum by 9th month or close to birth. So after birth, we examine the testis of the baby, whether both the testes are contained within the scrotum. If not, that is, one is present and the other is not, or both are absent, then we need to do an operation which is known as orchidopexy. You have to draw down the testis into the scrotum and ligate it, fix it. The scrotum is drawn down to the scrotum and the lower pole is fixed with the scrotal wall. This operation is known as the orchidopexy. Right? Now, can you see the genital cord? The two mesonephric grid. On the chest there is genital ring and the joining part is the genital cord. Can you see? Laterally there is the mesonephric tubule and in the midline there is paramesonephric tubule. These are going to what? Urogenital sinus that gives rise to the bladder and urethra. So by 5th week, from the dorsal wall of the gut and through the dorsal mesentery, the primordial germ cells are entering into the either cortex or ovary. Right? Then cortex or ovary is turned into either testis or ovary. This is done in case of ovary, it is 8 to 9th week, and in case of male testis, it is by 7th week. So 5th week is the primordial germ cells are coming. Then 7th week, the medulla is turning to testis, and in case of female, by 8 to 9th week, cortex is turning to ovary. Alright. Next comes the question of phenotypic sex, where the Mullerian degradation factor from the Sertoli cells and the sex steroids, that is testosterone and 5-hydroxytestosterone, will do the job. Right? So, we will describe the development of the testis by describing number 1, the chromosomal sex, the gonadal sex, next the histological differentiation, and next the phenotypic sex. Right? And following that, the descent of testis, causes of descent of testis, and the sites where the testis can go. In case of pig, the testis remains in the superficial perineal pouch. And in case of elephant, both the testis remains within the abdomen, that is intra-abdominal. In case of the marsupial, the testis moves to the anterior surface of the symphysis pubis. In case of marsupials, kangaroo, koala, for them the testis comes to the anterior surface of the symphysis pubis. So, this is for the marsupials. Within the abdomen, that remains in case of elephant, and in the superficial for the pig, it comes into the superficial perineal pouch. And the other important thing is the coming to the saphenous opening. Testis may come into the saphenous opening. So, without this knowledge, you cannot, after the delivery of a baby, birth of a baby, without the knowledge of where the testis could be found, you will be misdiagnosed a case, normal case, as an undescended testis. So, you must know the remnants of the gubernaculum and the tail of locule, so that you can identify where I can find the testis. Then you will be able to stamp this case as an undescended testis. Alright. Together with this, what we see in the baby room after the delivery, that is whether there is cleft lip and cleft palate. This is also very important. Alright. Okay. Thank you all.